I repeat, there are over 200 COVID-19 candidate vaccines in development, with about 2 dozen at clinical trial stage with human volunteers within the last 8 months. Below is a list of some vaccines in various stages of clinical trials. We have the list shown on the screen. We have quite a lot: US, China, United Kingdom, and for United Kingdom, the trial is in South Africa. You also have GSK, you have AstraZeneca, phase 3 of the test, and China approved for limited use on August 31st, 2020. Russia has also moved to phase 3, 2 weeks after it received an early approval. So we have all this. These are just a few of these, and the source is reference 9, New York Times. Support healthcare heroes. Pharmacists with other healthcare workers are in the forefront of fighting the disease, and many even contracted the disease in the course of discharging their duties. Despite all the challenges of COVID-19 pandemic, the pharma sector and our frontline healthcare heroes have risen to the challenges, holding the fort and marching courageously to victory. We expect to be in the promised land very soon. We salute the scientific efforts in pharmaceutical revolution. We also salute the efforts and courage of our frontline workers, our healthcare heroes. Nigeria's COVID-19 situation chart. Just to show you what we're going through in Nigeria, we have sample tested, 411,077 samples as at September 1st, 42,020 and 30 confirmed cases, 54,247 confirmed cases, 42,010 discharged, home, healthy, and 1,023 deaths. And we have about 36% of female, and again 64% of the male. So we have more of the male with confirmed disease infection, and that's the spike. That's the daily epidemic confirmed cases. Nigeria's response to COVID-19 has been swift and remains good so far, probably because of our experience with Ebola, 2 years ago. From the chart, it is evident that there is high recovery rate, and so far Nigeria is doing well. In conclusion, because we do not have a time, even though there are lots of controversies, it is good to commend the scientists who are involved in the studies. All the studies, observations, and the over 200 candidate vaccines happened within the last 8 months to show that the response from the pharmaceutical sector is indeed a revolution. Even with the herbal formulations and immune boosters, pharmaceutical revolution is the response. Healthcare heroes. I want to thank you for your attention. I love to acknowledge Dr. Longshola, Ms. Comfort Oga, and David Oyeride for being in my team. And the references are shown. Thank you very much. Thank you very much, the moderator. I'm through. Thank you very much for your attention and audience.
Dr. Femi, we appreciate your fruitful participation in the conference and your emphasis about how COVID-19 is a revolution, not evolution, which is related to our experience in our practice in the daily life. We have to make abrupt change in this life. Thank you. And any question? There is no question.
Oh, thank you very much.
Thank you.
Yes, thank you very much.
Thank you.
Thank you, Dr. Magda. Thank you, Dr. Femi. Now, the next speaker, Professor Dr. Sanjay Jashak. Professor Jashak is here. Professor Jashak.
Yeah, I'm there. So,
Welcome.
yeah,
Professor
thank
Jashak,
you.
Head of Department of Natural Product, National Institute of Pharmaceutical Education and Research, Punjab, India. Dr. Sanjay Shishak is affiliated to Department of Pharmaceutical Education, National Institute of Pharmaceutical Education and Research, where Dr. Jashak is currently working as professor and in charge in natural product. Dr. Jashak major research interests involve screening of plant extracts and their isolates for COX-2, COX-1 inhibitor activity, which may be helpful as the lead for the development of safer anti-inflammatory drugs with minimum side effects. The talk is about role of natural products and traditional medicine in fight against COVID-19. Dr. Jashak, you can start now.
Okay, good afternoon, good evening to all of you and greetings from India. So first of all, let me thank the organizers for inviting me to deliver lecture in this wonderful online conference, experience in its of own kind. So with this, I also thank Professor Umran and Professor Magda, the moderator of the session, for kind introduction. So may I allow to share my screen now? Am I audible? Hello.
Yes, Prof, you are a co-host. You can share
Yeah,
your screen now.
yeah, I will share my screen. Just a moment.
Platform and management team, stop sharing the screen from your side, please.
Yeah. So can I start now? Share the screen. Yes. No.
Please try again. Try again now.
Okay, just a minute. Just a minute, actually. Yeah. So is the screen visible to all of you? Is my screen visible? Hello.
Yes, it is visible.
Yeah.
You
So
can go on.
the topic of my talk today is role of natural products and traditional medicine in fight against COVID-19. So we know several speakers in this conference emphasize the role of herbal medicine. So my talk is related to that. So I have planned first of all, I want to just put this disclaimer. The views expressed in this webinar by me are personal and it do not necessarily represent the views of my institute. So I have divided my talk a little bit introduction and some phytoconstituents which have shown promise against the SARS-CoV virus as well as the SARS-CoV-2 virus in silico studies. And what is the Indian approach that we are taking forward to find a drug molecule or you can say formulation that I'm going to present. So the speakers have already talked about COVID-2 or this COVID-19 disease or SARS-CoV-2 virus. So as we speak globally, now as of 2nd September 2020, there have been over 25.6 million COVID cases throughout the world. That includes also more than 852,000 deaths. So the pandemic is growing at a large or a faster speed. So we really need to find effective treatment, including vaccines and the drugs, and also the role of these herbal drugs and their formulations is big here. So that is what I will be emphasizing. So as we speak here, the SARS-CoV and MERS-CoV are the two major causes of the severe pneumonia in humans that we are seeing. And we are all still wondering what is the exact origin of virus. That continues to remain as a mystery as on date. So the treatment as we are talking about, there is a lot of repurposing as we are from pharma field, we know we can do the repurposing. The well-known drugs which are studied for their safety efficacy, we can study for the new diseases, like we are talking about this COVID-19. So the earlier drugs for different diseases, viral diseases against HIV, herpes, hepatitis, influenza, SARS-CoV, MERS-CoV, and Ebola, they are now being repurposed for the treatment of COVID-19. Already mentioned by the earlier speakers, the effective drug remdesivir and favipiravir, they are now approved for the treatment of COVID-19 disease. But still, we have to find an effective treatment for this disease, and we are waiting for vaccine to be approved. So in India, large effort is going on. We are expecting the vaccine by the beginning of next year for sure in India. That is what the current status. But having said that, what is the role of our traditional medicine, whether I see India, China, or even Egypt? So we have very rich, ancient traditional systems of medicine. Why can't we use them to find some lead molecule that will be useful in treating this COVID-19 disease? So with this, I come to some phytoconstituents. So I don't want to just bombard all of you with the molecules, but certain compounds from different traditional medicines where we use mostly medicinal herbs. What are their constituents? They can be taken as lead molecules with respect to the screening by the in silico methods against the druggable targets of this SARS-CoV-2 virus. So the major druggable targets are 3 chymotrypsin-like protease, pepin-like proteases, we say PL-PRO, then the RNA-dependent RNA polymerases, and spike proteins. These are the 4 commonly used targets now, druggable targets, to find the leads for this SARS-CoV-2 virus and their viral proteins I just mentioned. So with that, you see several natural products, especially from the medicinal plant origin, as phytoconstituents I have mentioned. So we start with several, you see on this slide, are there of different types of secondary metabolites of plants. So the first one, betulonic acid, that I made in bold. So that showed inhibition of replication with sub-micromolar IC50 value. So betulonic acid is nothing but the oxidized derivative of betulonic acid, which is a commonly found pentacyclic triterpenoid in several plants, and green tea catechins. So they have actually their role in this inhibition of the virus that I have shown the structures on the slide, the catechin gallate and betulonic acid. So further on this slide, you see 2 interesting molecules I want to tell. Imetin, as all we know, very old, commonly used alkaloid to induce this vomiting, and also it is very good anti, you can say the agent which is used for several diseases like malaria and others. So that alkaloid shows a very good promise as antiviral agent, and it is definitely a molecule to be looked at. Apart from that, we have several diterpenoids, like here ferruginol, which is a ABA10 type diterpenoid, showed the inhibition of replication of the SARS-CoV. So this is the imetin, which is a pyridoisoquinoline type of alkaloid that is known to us since long time. And you can see on the right-hand side, which is a ABA10 type diterpenoid having the phenolic ring here, as it has a phenol group. So it is a meroterpene type of compound, which is a partly terpenoid, as you can see, an interesting molecule. Then we do have several other diterpenoids, like here mentioned one example. It shows inhibition of replication, and some flavonoids like luteolin, they have shown promise here, that are common ingredients in many herbal drugs. With that, we see this lycorin, which is the amyloidaceae type of alkaloids. In that, we have lycorin, which is a toxic molecule, but it showed very good IC50 values against several viral proteins. And the mycophenolate mofetil, as we know, is an immunosuppressant. So this is the compound also showed promise against several viral proteins that can be considered for further studies. This is the indolizidine alkaloid called lycorin and mycophenolate mofetil. So with that, the another example here, scutellarian, a flavonoid that shows a potent inhibition of 3-CL-Pro. that is 3-CL protease inhibition with this 0.86 micromole IC50 value, which is itself shows the simple flavonoid can be definitely considered, which is present in Scutellaria species. So apart from this, it brings to me a plant, Tylophora indica. We did a lot of studies on this. It is a source of very interesting alkaloids, and they show, you can see sub-micromolar on in nanomolar range IC50 value against this 3-CL Pro. So in our in silico studies that we have done recently, we have found several interesting compounds from few Indian medicinal plants like Tylophora indica. So we have these two compounds, tylophorin 7-methoxycryptofluorine. Now you see, this is the compound that I'm talking about, 7-methoxycryptofluorine, which is a phenanthrene, and that is attached to a quinolizidine. Whereas the tylophorin, which is also a phenanthrene nucleus attached to indolizidine. So you can see this interesting skeletons which are present, showing very effective inhibition of viral proteins can be considered. So this tylophorin from the Tylophora indica plant, which we use the aerial parts of this plant to treat several diseases, including the common flu. So with that, this brings me to several Indian medicinal plants, like here let us say Glycyrrhiza glabra, Clitoria ternatia, then Evolvulus alcinoides, Andrographis paniculata, and several Tulsi species, Ocimum species that we use in our traditional medicine like Ayurveda, where they have shown a lot of promise to have the activity against the SARS-CoV, as well as the cytokine storm that we see in this disease as the disease progresses. So we have several interesting medicinal plants that shows very potent anti-inflammatory properties that can be explored also as a solution to treat this COVID-19 infection. So with this, there are certain therapies mentioned in our traditional systems of medicine. We abbreviate them Ayush. So Government of India started a separate dedicated ministry called Ayush Ministry in the year 2014 to promote the research in this field, where the Ayush, I just explained, Ayush is Ayurveda, A stands for Ayurveda, Y is for yoga and naturopathy, U is for Unani, S for Siddha, and H homeopathy. So homeopathy we have adapted from Germany. So this completes 5 systems of medicine together called as Ayush in India. So we have several interesting medicinal plant for preventive and prophylactic treatment like Tinospora cordifolia, very important plant. That is lot of research is being done, and also this is one of the lead plant to find the treatment for COVID-19. Then Andrographis I mentioned in the previous slide. Then we do have certain formulations like Ayush 64 Ayurvedic formulation for the symptomatic management of COVID-19, already approved by the Ministry of Ayush, Government of India, in this at the COVID centers where we have the quarantine facilities. There these treatments are given also to the patients with the Ayurvedic formulations, and their clinical data and observations are being recorded. That will be really useful, as we have a country of 1.35 billion people. So definitely we need to look at our traditional system of medicine, and we have wonders there. We can really do the clinical research, and that is being undertaken right now, where we have several such formulations that can be used. With this, it brings me to the category of new drugs, new drug category approved by our main drug regulatory authority called as Drug Controller General of India. We call it as DCGI. So this category of phytopharmaceutical that was approved in 2015. So I will come to that in the next slide, but we are talking about paradigm shift. So while the paradigm has evolved in identifying one cause of a disease and one chemical entity to correct that cause, the traditional systems of medicine in many countries, cultures, which have used extract from natural sources, has by and large been ignored so far. But due to increasing debates and concerns over the decades, US FDA finally in 2005 recognized botanicals as the class of medicines which are plant-derived natural extracts and complex mixtures of compounds. where the botanical drugs are not necessarily purified chemical entities to treat the disease. So the same class of drug was adopted in India as phytopharmaceuticals in 2015, and several or a few, at least I can say, phytopharmaceutical products have under different stages of development currently in India for different diseases, including COVID-19. So what is this phytopharmaceutical that our Drug Regulatory Authority recently approved in 2015? It is a drug that is defined as a purified and standardized fraction with defined minimum 4 bioactive or phytochemical compounds, qualitatively and quantitatively assessed of an extract of a plant or its part for internal or external use of human beings or animals for diagnosis, treatment, mitigation, prevention of any disease or disorder, but it does not include administration by parenteral route. So phytopharmaceutical drug will be regulated like that of our conventional allopathic drugs or new chemical entity based drug. So to develop such a phytopharmaceutical, we need to have certain basic requirements like available information on the plant, formulation and route of administration, dosage, therapeutic class for which it is indicated, and the claims to be made for this phytopharmaceutical, and supportive information from literature on safety and efficacy, and human or clinical pharmacological information. So we need to generate the data on identification, authentication, source of the plant used for extraction and fractionation, process for extraction and subsequent fractionation and purification. Here we can have the process patent for the enriched fraction that we are talking about, which is standardized to actually 4 bioactive constituents or markers at least, that we can have the patent here. Then the formulation details of phytopharmaceutical drug, and then manufacturing process of the formulation and stability data. All this has to be provided, and then we can submit the application to our drug regulatory authority, which takes minimum 1 to 2 years to give the approval. But recently, in the month of June 2020, among this pandemic, our DCGI has approved the clinical trial for the extracts of the plant Cocculus hirsutus. In Hindi, it is called as Patal Garudi. That is spearheaded by the efforts or intervention, you can say, of academia and industry, Sun Pharma of India, and the ICGB, that is our Council of Scientific and Industrial Research Labs in Delhi, and this Indian Institute of Integrative Medicine in Jammu, where they have actually launched this phytopharmaceutical. An interesting story is that this express approval is given by our Drug Regulatory Authority for the treatment of COVID-19 of this phytopharmaceutical. This was earlier they were developing as the therapy against dengue virus. Now you can see dengue virus and coronavirus, as we are talking about SARS-CoV-2, they have a lot of similarities. And on that basis, DCGI has given the express approval for the clinical trial of this particular phytopharmaceutical. So similar efforts we are doing at our department, like I told you, Tylophora indica and Vitex negundo, then Muraya konegi, where we are actually developing a phytopharmaceutical on the lines as it is already approved by this particular example. And this is the new information, new category of drug that I want to tell all of you who are attending this particular conference online, that we have a new category of drug where we really can promote these herbal drugs and their formulations by the name phytopharmaceuticals. So with this little bit, I want to just spend 2 minutes on our department where I work since last 22 years. Now I'm working as professor and head of this department. So we have several areas of research, including the infectious diseases, where we have the viral diseases, leishmaniasis and tuberculosis. Apart from that, we work on the non-communicable or lifestyle diseases like the inflammation, then the obesity, cancer, and then standardization of herbal drugs and formulations, development of phytopharmaceuticals, nutraceuticals. So we have already some mega projects where we are developing the phytopharmaceuticals as well as the herbal formulations for different diseases like escape pathogens, as well as for the, let us say, obesity and related areas like the sea buckthorn. where we are already doing these formulations, and these are the details there. And all this, we do a lot of dissemination of the information by organizing international conferences. Like our department, since last 10 years, we are organizing, or 12 years, every 2 years, this international conference on drug discovery from natural products and traditional medicines in India. And we do have a lot of collaborations also with the TCM, traditional Chinese medicine, that in 2018 we initiated by starting with this conference. That is the recent one I just want to show. And with this period, we have already published several patents, publications in the international journals of repute, and even the monographs on Indian medicinal plants, and the monographs on herbal formulations and Ayurvedic formulations like Arishtas and Asavas, and several other efforts we are doing in promoting the traditional systems of medicine in India. With this, I thank you all. Before that, I am talking about where I'm sitting right now. I'm sitting here. This is my cabin here in the Department of Natural Products. This block is for the Department of Natural Products in our institute, and we do have a dedicated facility, Natural Products Field Laboratory, where we have a lot of processing and extraction facilities on large and pilot and lab and pilot scale to do the research on this medicinal plant. So with this, I thank you all for patiently listening to my talk, and I'll be happy to take a few questions if you have. Thank you, organizers. Thank you so much.
Thank you very much, Professor Rashak, for this valuable and interesting talk, and also valuable and interesting area of research. Thank you very much. Any question?
Thank you.
Please, Dr. Gamal, I'd like to ask Dr. Sanjay about
please.
his big number of alkaloids containing active constituent acting as protease inhibitor. So I would like to ask you if you make a correlation between the chemical structure and mechanism of action. You have many ketone group, and you have many methoxyl group, you have many phenolic group.
Exactly. Yeah, so see, when we talk about this correlation, these studies which are being done right now in vitro. Okay, so with the first in silico, you can say leads, where we have seen the binding of these particular compounds, let's say phenolic compounds. They do bind the phenolic groups, bind with the viral proteins, like we are talking about 3-chymotrypsin-like protease, 3CL-Pro. So most of the studies are against this 3CL-Pro of SARS-CoV-2 virus. So then we have to take it to the in vitro enzyme assay and then the cell-based assay. So in that, definitely, when it comes to the in vitro studies, we can have the mechanism because we know the binding mode. Let us say whether you have the phenolic OH groups, or let us say the carboxylic groups or the amide groups.
There is a glutamine, aspartate, and asparagine amino acid, which is very essential amino acid for protease enzyme. So this is the second stage for your study.
Exactly. So, but in vitro studies and in silico studies, you can try to tell the mechanism and then take your active molecules towards the cell-based assay. Okay. That is the ultimate one where you can see the promise of that. And once you have the activity in cell-based assay, you can go for the clinical trial.
Okay. Hope all the wishes for you.
Right.
Thank you.
Thank you. Thank you.
Thank you, Professor Rashak. Thank you, Professor Magda. Another question? Thank you for all. The next speaker, Professor Dr. Magda Ismail.
Yeah, thank you, Dr. Gamal. Now we will move to the fourth talk introduced by Professor Mohammed Mabrouk Abul Wafa, former chairman of National Organization for Research and the Control of Biological. His talk will be on vaccine development, historical analysis, and how to accelerate. Please, Dr. Mohammed, if you are here, please start.
Thanks very much for Dr. Ayman Sharif and Dr. Samar for inviting me to participate in this interesting conference, as well as my great thanks to Dr. Gamal and Dr. Magda Ismail, the chairperson for this session. Let me share my presentation. vaccine development, historical analysis, and how to accelerate. Vaccine development. Vaccines development and use are among the greatest successes in the history of public health, and still the most effective public health measures available. The use of vaccines have resulted in eradication of smallpox, near eradication of polio, prevention of millions of deaths each year. Stages of vaccine development. The first stage, the first vaccines. The second stage, the second renaissance of vaccine development. The third stage, the latest renaissance of vaccine development. The first vaccines, they are called conventional or traditional vaccines. In 1796, Edward Jenner introduced the term vaccines for the first time. This scientist succeeded to protect against smallpox infection by using cowpox-infected materials for immunization. Louis Pasteur refined Jenner's work through using weakened forms of a pathogen to promote immunity against virulent forms of the pathogens. Pasteur principles of vaccination are isolation, inactivation, and injection. This approach constitutes the basis for many of the first vaccines. Advances in vaccine propagation, in virus propagation, and cell culture facilitated the development of methods for attenuating viral vaccines. The second half of the 20th century is considered a golden age of vaccine development, and several vaccines were developed during this period. These included polio, measles, mumps, rubella, and varicella. The first vaccine development based on using the entire pathogen in either a killed form or live attenuated form. Vaccines developed by this way were termed conventional traditional vaccines. The use of conventional traditional vaccine led to drastic reduction of many diseases. For example, measles, mumps, rubella, diphtheria, tetanus, pertussis. Limitation of conventional traditional vaccines, in vitro culturing difficulty of some infectious agent, this as an limitation, undesirable immune responses produced by certain attenuated pathogens. Conventional traditional vaccines were impractical for pathogens that are either exhibit antigenic hypervariability, for example, HIV-1, HCV, or those exacerbate diseases upon vaccination, for example, RSV, respiratory syncytial virus, and the dengue virus, or those have an intracellular phase, for example, tuberculosis and the malaria. The second renaissance of vaccine development, this was based on the development of subunit vaccines. Subunit vaccines use only part or parts of the infectious agent, such as certain protein or peptides or carbohydrate antigens to induce an immune response and protect against infection. The criteria of choosing subunit vaccines as a candidate, the targeted component of the pathogen should be abundant and conserved, can be seen by the immune system, and is capable to induce a protective immune response. Subunit vaccines can be improved by optimization of delivery systems, tuning the size of the pathogen particulate component, targeting specific immune cells, and adjuvanting the vaccine. There are two ways to construct a subunit vaccine. First, by isolating a specific protein from the infectious agent. Second, by molecular techniques through expressing an antigen in a vector. Examples of subunit vaccine, a cellular pertussis vaccine. In this vaccine, we use the toxins of Bordetella pertussis, and we carried out detoxification either by chemically or by applying molecular genetic techniques. Another example is the Hep vaccine. which is based on hepatitis B surface antigen, which is produced in yeast cells. Another example, bivalent and quadrivalent human papilloma vaccine, HPV, which is consists of virus-like particles, VLPS, which is self-assembling of L1 structural protein copies of the HPV virion. Conjugate vaccines. Bacterial capsular polysaccharides can be conjugated to a carrier protein or to carrier proteins. This makes polysaccharide antigens to be T-cell dependent rather than non-T-cell dependent, and thus immunogenic for infants. This resulted in the development and the licensure of Haemophilus influenzae type B, meningococcus A, C, W, and Y, and the pneumococcus vaccines. The latest renaissance vaccine development, inactivation and attenuation fail to induce sterilizing immunity when applied to certain pathogens. For example, pathogens that exhibit antigenic hypervariability, for example, influenza, dengue, HIV-1, HCV, or those their conventional vaccines exacerbate diseases, for example, RSV, respiratory syncytial virus, dengue virus, or pathogens which lead to persistent or latent infections or have an intracellular phase, for example, HIV-1, hepatitis C virus, cytomegalovirus, herpes simplex, mycobacterium tuberculosis, and malaria. Accordingly, viable vaccines are needed to induce protective immune responses beyond the mechanisms evolved by nature. New vaccine development technologies are based on either antigen discovery and development technologies. This is the first approach to identify antigens that are able to induce sterilizing antibody responses and generate T-cell, such as reverse vaccinology, genomic-based antigen discovery, which is called antigenome analysis, structural vaccinology, and synthetic biology. The second approach, vaccine delivery technologies such as viral vector-based vaccines, nucleic acid vaccines, DNA or mRNA vaccines, virus-like particles, liposomes. Regarding the first approach, which is identifying the antigen to be used as a vaccine candidate, the first parameter, reverse vaccinology. Reverse vaccinology involves screening the entire genome of an infectious agent to identify gene or genes encoding proteins which with appropriate characters of good vaccine targets. They that are surface exposed, secreted, highly conserved among strain. Ideal candidates are selected, then expressed and used to immunize lab animals for evaluation of their immunogenicity and protection capability. Limitation of reverse vaccinology is their inability to identify non-protein antigens, such as polysaccharide, which is a component of many successful vaccines. Application of reverse vaccinology. Reverse vaccinology is used for development and the licensure of vaccines against meningococcus B, MenB. Also vaccine candidates against several bacteria, including Staph aureus, group A Streptococcus, Streptococcus pneumoniae, chlamydia, are in advanced preclinical and clinical studies. The second parameter, genomic-based antigen discovery, which is called antigenome analysis. This technology involves interrogation of the comprehensive antigenic repertoire of genetically expressed antigens, then screening for their immunogenicity. Mass spectrometry helps in identification and the quantification of antigens on the surface of many bacteria. The second parameter, structural vaccinology. Structural vaccinology involves rational engineering of immunogen. Antigenic epitopes are identified based on the protein amino acid sequences and then or the resulting secondary and tertiary structures. The concept of structural vaccinology and efficient immune response needs only recognition of a single or multiple selected epitopes, but not the whole antigenic protein. This is mostly sufficient to induce protective immunity. The component of structural vaccinology, two distinct components are recognized. The first, conformational stabilization. The second, using scaffolds to target specific epitopes. Application of structural vaccinology, while reverse vaccinology helps in vaccine development in case of bacteria and parasites, structural vaccinology will have an impact in developing vaccine against viral infections. The first, conformational stabilization. This is represented by fusion glycoprotein or if glycoprotein of respiratory syncytial virus. This glycoprotein occurs in two conformations. The first, pre-fusion protein, the second, post-fusion protein. Both are immunogenic and induce neutralizing antibodies formation. Stable pre-fusion F antigens is much more superior immunogenicity relative to the post-fusion antigen. Second, targeting a specific epitope using scaffolds, which is called reverse engineering on vaccine. Reverse engineering on vaccine depends on identification of broadly neutralizing antibody. Second, determination of the structure of the binding sites of such a broadly neutralizing antibody using crystallographic methods. Third, mimicking the epitopic binding sites of such a broadly neutralizing antibody on carrier protein scaffolds to act as immunogen. For respiratory syncytial virus, computationally designed immunogens mimicking the binding site for an RSV neutralizing monoclonal antibodies have successfully elicited RSV specific neutralizing antibodies in monkeys. The last one, for recognizing antigen to be used as a vaccine candidate, which is synthetic biology. This involves gene synthesis in the laboratory that can be subsequently cloned and expressed in an appropriate host. This figure shows the reverse engineering of vaccine. You need to determine the broadly neutralizing antibody from infected patient. Then you need to molecular characterize the binding between broadly neutralizing antibody and the antigen. Third, you need to construct the immunogen depending on the molecular characterization. Last, you need to add adjuvant or adjuvanting the immunogen. The immunogen will be constructing using scaffolding protein. The second approach for new vaccine development is based on delivery technologies in vaccine development. The first, viral vector-based vaccines. Many viruses can be used as a vector for delivering certain antigens, which included adenoviruses, some species of parvoviruses, togaviruses, some strains or certain strains, paramyxoviruses, certain strains, rhabdoviruses, such as vesicular stomatitis virus, and the poxviruses such as modified vaccinia Ankara virus, MVA. The second, nucleic acid vaccine. Nucleic acid-based technologies employs either plasmid DNA encoding antigen genetic materials or RNA as mRNA or viral replicon. Upon their cellular uptake and expression, the produced protein can elicit humoral as well as cell-mediated immune responses. DNA vaccine.
Excuse me, Professor, the time is about to finish. We need you to be fast, more.
Okay.
Thank you.
DNA vaccine and RNA vaccine show some advantages over other delivery technologies, especially RNA vaccines. RNA vaccines either non-replicating mRNA or self-amplifying mRNA. This table summarizes all the properties of discussed vaccine technologies, either viral vector-based vaccines, DNA vaccines, RNA vaccines. Virus vaccine platforms. Platforms technologies allow multiple vaccines to be more rapidly produced from a single system. These technologies is defined as a platform if an identical mechanism, device, delivery vector, cell line can be employed for various vaccine targets. These included viral vectors vaccines, nucleic acid vaccines, either DNA or mRNA, cell line that can be employed, that can be coupled to viral expression system platform, virus-like particle platform, liposome platforms. This table shows the spectrum of platforms technologies, either viral vectors, cell line, viral expression coupled system, nucleic acid, virus-like particles, liposome platform. This figure shows the modularization of the target epitopes using any target epitope from either bacteria, virus, or parasites to be inserted in a platform base, either virus-like particles or liposomes, to give a modular vaccine. This table shows the different vaccine technology platforms until August 2020 for in case of COVID-19. There are about 14 vaccines in phase 2 and phase 3 clinical trials. And these platforms included the recently developed platforms as well as the traditional ones depending on inactivation and attenuation. Conclusion. Candidate vaccine development can be accelerated with the use of vaccine platform technologies. A platform vaccine production facility could be switched relatively easily to production of various vaccines. The modular nature of platforms means that regulatory approval can be expedited as these technologies become more commonplace. Approval can be accelerated if regulators have previously authorized similar products before. Labeling a vaccine technology as a platform heighten attention by the media, investors, funders, and the public health leaders. Platform vaccine technologies have the potential to minimize risks associated with emerging infectious diseases. Traditional vaccine development should and will continue to play a critical role in vaccinology alongside platform-based approaches. Thank you very much.
Thank you, Dr. Mabrouk. I am very grateful to you for your distinguished talk, which is in the core of our conference, the core subject of our conference. So deep thanks from me to you and for being committed to the time limit. Thank you very much. Any questions? There is no questions.
Okay.
Now we arrived to the final talk by Dr. Islam Anan, lecturer of the pharmacoeconomics and the pharmacoepidemiology, MIU and future. He is teaching in all these places. He will present his talk regarding the role of health policy and economics in response to COVID-19. Hello, Dr. Islam. Are you here?
Hello, Dr. Magda. How are you?
Oh, welcome. You can start your talk now. The floor is yours.
Thank you, Dr. Magda. So first of all, I would like to thank the committee for the opportunity. I would like to thank you all for being here. I know it's Thursday and it's in the afternoon. I will try to be as quick as possible, and I will be talking in brief about the role of health policy and health economics in response to COVID-19.
Dr. Islam, take your time, no problem.
Thank you.
Okay.
And mainly it's because it's a new science. Health economics we implemented health economics in 1992. That was the first time to implement it, and health policy as well in regards to health economics and in regards to pharmacoepidemiology implementation was new to the world. So how we actually responded, being a pharmacoeconomist, pharmacoepidemiologist, and health policy specialist to COVID, will be going through very quickly the Egyptian case study, and we will be looking as well on the African countries. So without further ado, that's my conflict of interest. And just a very quick glimpse at the history of pandemics and the death toll, that coronavirus, the COVID-19 disease, is not the first pandemic to face a population and face the history of mankind. We've been subjected to a lot of pandemics. It's actually ranked number 12 in terms of how it's actually affecting deaths. But we hope not, but it's forecasted to be actually in this place, to be along with the Asian flu in terms of death toll, to exceed 1 million deaths. So how can healthcare system solve any pandemic? So here we'll be talking from idealistic perspective about healthcare systems. And throughout all the different healthcare systems, we'll find that the world faced the same pandemic. We got the same obstacles of the healthcare capacity, the number of beds, the awareness amongst the public sector. However, what was different is the different approaches. What have we done different in terms of country to approach this problem? So we all the time say in conferences and in universities that health policy obstacles are all the time the same. The UK system got a waiting list, which is more or less the same waiting list we are facing in Egypt. We got deficit in budget sometimes in Germany, which was faced by UAE and so on. So to make the reform and to face the pandemic, you need to have a very good health policy, and you need to have a very good health economics in place. So in health policies, they put certain parameters in terms of handling the COVID condition, which is you need to have an A, B, C, D, E. A stands for access, and by access, we mean that patients with COVID can actually access healthcare facilities and get their treatment, which is a very important aspect. And we've seen that not all the patients were able to access, in some countries, healthcare facilities, get their treatments during the time of surge in the first couple of months in the pandemic. B stands for best quality, which is what will be the protocol and health quality care that we're going to be giving to the COVID-19 patients. And C stands for cost, whether the country will be able to provide all the preventive measures and all the medications and the protocol with a reasonable cost, a cost-effective measure, and at the same time to be providing the protocols to the patients for free. D stands for differentiated. We're not going to be able to make one size fits all solution for all countries. So we need to have a certain solution for Kenya, a certain one for Algeria, another one for Egypt, and so on. And finally, that the system should be effective. So if I'm giving a treatment for a patient, he should be treated. That's why it's an effective system. So that's an A, B, C, D, E of health policy. The country managed to do the A, B, C, D, and E, managed to get rid of the first wave quickly and managed to get with the least damage possible. And if you can do an A, B, C, D, and E, you manage to have equity among your citizens and population and solidarity. So that's health policy. And on the other side, you need to manage the economics of the system, which is you got low supplies, you got limited budgets. No one forecasted a budget for COVID-19 in 2019. It was a shock for us. It was new. We had to get budgets from other ministries. We had to get budgets out of the Ministry of Health and Ministry of Finance. And at the same time, we got very high demand of healthcare services. So it's not an equilibrium in terms of system. And putting and adding to that, that we don't have an efficient system. We have a lot of wastage among all the disposables that we are using. We got a lot of wastage among the use of medications as well. So if you got the health economics, health policy in place, you got a way to actually mitigate the second wave, for example, of COVID. If we have learned something from the first wave is how to mitigate the second one. So to manage any pandemic, we need to, for this slide, to achieve it, to achieve the A, B, C, D, and E, and to manage the economics of the pandemic, to manage the resources, to have enough money, and to provide. So that's the idealistic scenarios. And actually, no country managed to actually get to the idealistic scenario. However, we got a lot of defects in A, B, C, D, and E. We got a lot of defects in managing the resources and the economics. And actually, if you have a proactive disaster management system, which is a big part of health policy, you will be able to mitigate not only COVID, any future pandemic. And if you got a force majeure like this pandemic, and you got 4 stages of disaster management. The stage of mitigation, which is how to be proactive that you're not going to be facing any pandemic. That's a phase of mitigation. If you are faced with a pandemic, how you can be prepared upfront before this pandemic hit the population. And if you are not prepared, do you have any measures to respond to the pandemic itself? And finally, if you got hit badly in terms of mortalities, in terms of incidents, how you can actually recover the system. These are the main 4 stages. And of course, because it's not a theoretical lecture and we're not going to be going through methodologies, these 4 pillars got a lot of science behind for each one of them. And of course, if you got preparedness and planning, the time and the pace and the crisis are not going to be as huge if you got the 4 pillars in place. The most problem is you don't have mitigation, you don't have preparation. We got faced suddenly with a COVID-19. We tried to respond, some failed to respond, some got it successfully, and we are trying to recover. What we'd like to do now is we'd like to have a mitigation system and a preparation system for any upcoming pandemic, not necessarily the second wave of COVID. And in that, countries in response to COVID, they do what is called is DIKW pyramid, which is the data to wisdom pyramid. It's how you can actually interpret any data coming from COVID, the incidents, the mortalities, and so on, and make this data as information to knowledge, and then take a wisdom decision. To give you a very quick example, data is a signal, information is how you can actually interpret the signal or the raw data. Knowledge, if you know what to do with the data, with the information, and wisdom is the integrated actionable decision taken, applied based on the knowledge. To give you a very quick example, even outside COVID, if I tell you 190, it got no sense because it's just data. If I told you that it's a systolic blood pressure measurement, it's an information now. You know what does it mean, 190? And if I tell you if it's high or low, if you know it's high, you got the knowledge. In your opinion, should we treat this patient? Should we return him back to be controlled? If you took this management decision, you got the wisdom. It's exactly the same story if we are talking about the figures and numbers of COVID. We take these raw figures and we translate it into information till we get to the wisdom part. You can see here, there's just raw figures. If you, for example, you're gonna divide the total recorded death over the total diagnosed and registered cases, it will give you a figure which is completely different if you actually divide the total recorded deaths on the total number of population, for example. So you got a lot of interpretation of data. The context of our lecture is not concerning that, but just to give you a very glimpse that we can use any type of data here to generate tons and thousands of information, tips that we can use in wisdom. For example, should we look at active cases or should we look at the total cases? Should we look at total recoveries and so on? So just to give you one example, we only rely on our models on prevalence. And for prevalence, what it means living with a disease, we'll be talking about the 20,000 patients in Egypt, not the 100. And even if the figure itself is not 100% correct, we need to work on the active cases. And this will actually take us to how much information do we actually need? So because of the data of incidence of all COVID patients, starting from China, Germany, States, Russia, African countries, is not 100% correct because it's actually relevant to the disease itself. We know that 80% of the patients are not going to be having symptoms that we can actually see on ourselves, and it needs hospitalization. So countries made two things in terms of health policies. They've done a reactive testing protocol for COVID or a proactive testing protocol for COVID. The reactive testing, it means that we're not going to be doing a PCR unless the patient got acute respiratory symptoms requiring him to go to the hospital. Proactive testing, it's like the UAE, it's like South Africa, it's like Saudi, it's like the States, Germany. They are doing mass random PCR screening so they can predict the number of active cases in the country, which is the most important figure. So imagine that from all the figures that we are seeing every day, that is the most important figure that we are concerned with, and which is a figure most of us just skip it while reading the daily reports. So moving from reactive to proactive, it means a lot of money. It's very expensive to go for a proactive testing. However, that's the only way if you'd like to have a good information about the number of active cases in a country. And you will find that the number of tests worldwide is relevant with the number of incidents and the number of infections and active cases. You will see that China got the highest number of total tests done. We are talking about 90 million tests. USA, India, Russia, UK, and Brazil, not a single African country. But if we look at the African countries, we've got South Africa at the top of number of tests done with 3.7 million, around 4 million, Morocco, Ethiopia, and Kenya. So the more tests you're doing, the more information you got about the active cases, and the more accurate your health policy will be in terms of preparation to the next wave. How many beds I need, how many ventilators I would like to be inside the hospitals, which govern rates I need to focus on during second wave. And you can see a linear correlation between the number of tests and number of cases. The more the tests, the more the cases. It's very obvious. The more you're testing people in the streets, the more cases you're gonna actually get, the more accurate you can find an information. And that's gonna help, that's the only way it will help in building a forecast model in terms of economic and pharmacoepidemiology models. We work with many models. If the amount of data and amount of knowledge is actually very high, and that's taking us to a knowledge pillar, you can do a simple SIR model, which is susceptible people, infectious and recovered, and just put in the model and calculate. Of course, because you don't have that knowledge, you can add recovered, it's very important, and work with an SEIR model. And because we depend more on deaths, we go for a SEIRD model and adding the dead pillar in the model itself. So if we have a susceptible people, because I'm doing a lot of tests, exposed, infectious, recovered, and death, I can build a model like that. This model, and it applies on all pandemics and all infectious diseases. It means that if I got a population of 1 million, imagine that if a country got only a population of 1 million, regardless it's 100 million, it's 50 million, it doesn't mean that all the citizens or all the population will get infected. You can see here that that's from the Ebola, for example. You got a susceptible, can be the full population are susceptible. However, the exposed for infection are around 30%, infected was around 25%, and once you got recovered cases above the infectious cases, you know that your curve of the infected, which is the yellow one, will start to decline. And that's how we managed to draw a curve for Egypt. So we've managed to do a forecast model that was issued in May 2020. And we expected that we're gonna reach the peak on the 1st of July. And we were talking about the death curve because we were not actually aware about the number of tests. We're doing limited testing. We're not doing a proactive health policy in terms of testing. So we were depending more on the number of deaths. and we were multiplying the number of deaths times 3. And in the 1st of July, we were accurate at 97%, and the total model got an accuracy of 93% so far since May 2020. We predicted till this day that we're gonna reach around, the first wave will reach around 5,800 deaths to 6,000 deaths. We are almost 5,400. We wish that our model were not gonna be true, and we close the deaths today before tomorrow, because every single dead person count. Of course, you will find here that there are some elevation again and incremental growth in terms of numbers, and that's because we reopened the economy on the 27th of July, and that's why we're expecting this fluctuation in figures, which is actually happening right now, and we can see it. We were getting in the SAIRD model the kind of activity forecast from Google Maps. If you know Google Maps, it sends a monthly report, on a bi-weekly report, sorry, on amount of activities outside homes. So it can track everyone got an Google Maps application on his mobile. We can trace him if he got outside of his place. You can see this line, this flat line, that's staying at the residency. Any curve, it means that here Egyptians are not staying at home. They are in the street. The more they are in the street, the more infections. And we can do some correlation exercise and end up with a chart. So moving to our last section for today's session is how can we actually mitigate the second wave? So the question is not whether we're gonna have a second wave or not. The question is when we're gonna have the second wave. In all pandemics and in all infections, we got waves. Luckily, that most viruses come second wave of it, a very light one, not as strong as the first wave. So health economics, it helps in prioritization of spending in terms of pandemics. But you need epidemiology profile, what we were just discussing. It's very important. Health economics are not going to be of value unless you got very good overview over the prevalence, over the how many active cases I got, then the clinical and economic burden, the opportunity cost. I'm going to illustrate it in a bit. Applying a political, P stands for political, economic, societal, technological model, the PEST model, which we call an HTA, Health Technology Assessment. That will make you able to allocate the right resources, working on the right process, and end the pandemic, the right outcome. And as you know, if we got like almost 862K deaths, almost 6.8 active cases, we spent so far $4 trillion US. So the opportunity cost is a health policy gonna make, whether I go for total lockdown, R&D, repurposing of drugs, whether I should have an innovative pipelines. Should I actually make a customized solution, or should I make Sweden model? Should I go for UK model? Should I go for my own model? So in the next slides, I will show you what countries have been doing and our recommended second wave mitigation for Egypt. The only country focusing on health economics of COVID is actually China. So China is doing all the time the impact of COVID-19 on the economy in terms of direct and indirect. And as you can see, the indirect is huge. We're talking about 380 billions. And the direct medical, what they are actually spending on medicine, on medications, on hospital, is 0.6 billions. As you can see, and I guess this will be a turnaround on the healthcare industry worldwide, is we need to allocate more money to healthcare. As you can see, a single problem, health problem, which is 0.6 billions in terms of cost, made tremendous loss of 380 billions USD only in 3 months in China. Imagine this figure now. So it's very important that we focus on investing more in healthcare, getting more budgets, and mitigate second waves. So far, we are expecting 350 million jobs lost, 270 million starvations, 12K poverty deaths every day, based on the global Oxfam report. So what should be the health policy to avoid the second wave? The WHO got a nice model called the VORGES, or the 4S model, which is space, staff, supplies, and system. If you got the 4S in place and ready, you are in a good preparation for any waves, whether it's COVID or even a new pandemic. For space, it means that you got estimated capacity for acute and intensive care. You got to develop criteria for the designated hospitals. You can regulate the patient flow. You can postpone all the elective surgeries and accelerate discharge because you need more space. You need to optimize the delivery platforms and purchase. If you don't have enough space, you can purchase it from the private sector. Repurpose non-medical facilities, get archiving rooms and make them ICU rooms if you can. Staff, in terms of quantity, you need more doctors, so you need to have plan B. And it's a very important point because we need to incentivize our healthcare professionals. They need to be valued in terms of monetary value and in terms of appreciation as well. So we can retain them and not to see migration from or expelling from inside a country to another. And in terms of quality, we need to upskill all the healthcare workers during the pandemic, ensure safety and protection, and take care of mental health, which is a very important point. Supplies, you can go for a unified purchase mechanism. And in Egypt, we got a very good example of that we have in place what we call Unified Purchase Association, UPA, and Hayat al-Shura al-Muwahhad, and they are actually the one purchasing for everything. So in terms of pandemic, it's always better to have a universal purchaser, not to segment or to make multiple channels for purchase. So you can actually control the counterfeit. You don't have a lot of counterfeit and you can control the price.
Dr.
You can identify
Dr. Islam,
Yes.
Islam, we are about to finish because your time is finished.
Yes, that's actually my last slide. The supplies,
We
ensure
give
and
you
assure.
extra time because you have already answered all questions coming to you inside your lecture. Okay, go
And I
on.
have 5 minutes more. If I
minutes.
can have 5 minutes more, it will be enough.
Yes,
Okay.
please, Prof. Magda, please, we would
Okay.
like to listen to Prof. Islam.
Okay.
Thank you.
And also to
So-
answer some of the written question in the chat box after that.
Okay, perfect. Thank you. Thank you for generosity and thank you for the 5 minutes. So finally, ensure emergency mechanism for any emergency pandemic, assure the products comply with the technical specs, and put a defined criteria. For the system, now it's very important to have a good supply chain system and to do a centralization of decisions. As we seen in the first pandemic, it was a Ministry of Health decision, then it was a Prime Minister decision, and now we can see that all the presidents all over the world are the one taking responsibility of the system and for COVID. So that's need to be planned and defined for any future pandemic. Should I go for R&D or just production of repurposed drug? The process, lockdown scenarios, having a hotline in place, awareness campaigns, and establish a public-private partnership. So these will summarize for Egypt specifically 10 steps to mitigate the risk of wave 2, increase number of tests, making it proactive, apply medical registry for all home quarantine, doing quantitative assessment, not to open all the economy at once, but just to assess every 21 days, expand the healthcare and pharmaceutical capacities, including ICU beds, for example, incentivize healthcare workers, it's a very important point, and make a reverse lockdown. Each governorate should be managed on its own. So I know that a single governorate got high incidence of cases versus 0 cases in other governorates that I can actually open for tourism, for example. Oblige large businesses and factories to take responsibility and CSR and pay for the protection of their employees, especially that we are approaching school days. Increase capacity of public transportation, enhance digital infrastructure, and finally, launch massive awareness campaigns. So we can have mitigation, preparation, response, and recovery. And in terms of equality is not that good, equity it's better, but in terms of pandemics, we need to have access, just remove all the barriers. And that's how health policy and health economics are in place. Thank you, and very thank you for the extra 5 minutes. I don't know whether I got some time to answer some questions.
Yeah, can I, Dr. Islam?
Yes, sure.
Yeah, this is Professor Sanjay from India.
Hello,
So,
hello, Professor.
yeah, very informative talk. I have seen you have been talking about wave 2. So, can you really tell all of us what are those indicators that really tells us now we have wave 2, but in India right now we are not able to decide. We still think it is wave 1 only going on. So, what are those parameters, indicators that tell us now wave 2 is there in place by looking at the disaster management and preparedness, as you rightly talked about the healthcare systems and the planning.
Thank you, Professor. It's a very good point. And all the actually countries are talking right now about it. So whether we are facing some relapse in wave 1 or it's a beginning of wave 2. So the indicators are, first of all, we look at the first wave start point, which was actually in winter. We look at the fact that all pandemics, they come in waves, and they look at the gross in terms of the end of wave 1. So if we do have a fluctuation as of 20% up to 30% of figures, it means that you're still in the first wave, and the first wave is about to finish, but just you need to control more the measures. But if the numbers are more than 30%, and you can see that death is actually elevating not only the cases, because that's the most important indicator. The wave will start with a high number of deaths, but if there's a huge increase in cases, but still the number of deaths, the mortality rate is decreasing, this is just the normal end of any wave. It's ending the wave. If you got an increase more than 30% in mortalities, it means that the wave 2 is actually connected to wave 1, which is the worst case scenario. And that's the scenario of Dr. Fauci in the States, who said that we'll actually connect wave 2 with wave 1. There is no break points. There's no break period between the two waves. The WHO was actually supporting as well the same scenarios, that we're going to be connecting waves throughout the years, and other scenarios which I actually optimistic about and looking at it, that we're going to have a break period, and the wave 2 will start with high number of mortalities again, but not as much as the wave 1. It will be like 50% less, because we do have a good preparation for the healthcare systems. And actually we can see that the viciously of the virus itself is beginning to be less than the beginning of the wave. So mainly to cut the long story short, mortalities are the most important indicator for the start of the wave 2.
Yeah, so luckily I can tell you, in India, we have now approached 75% recovery rate, and mortality is not that much. So let's see, it's a very good answers. Thank you for your response. Just a quick question as with respect to testing. So testing that we have to do aggressively, no doubt, as you have mentioned, but there is a lot of confusion, like rapid test, PCR test, RT-PCR test, and a CAT scan of the chest.
Yes.
So what do you suggest, which is the best test? Because we are getting a lot of false positive and negatives with the rapid test. So, and
Yeah,
RT-PCR takes a lot of time to come. So this is really a challenge when it comes to the testing, as we are still seeing in India. So
yeah, and it's exactly the same in Egypt. So we do have delayed in the PCR results, and we do have as well not enough tests for all the people. So we depend more on X-rays, we depend more on imaging, which is more than enough, because you don't wait till the results of the PCR are here to start treating. You just immediately treat as if it's COVID, and most probably it will be COVID, and you can do a PCR or not. The most important thing for us, being from epidemiology and from health policy perspective, is I need the record of the patient who actually did not do a PCR, however, was treated immediately based on imaging. The most problematic thing is we don't have the records. So countries are treating immediately, they don't do PCR, and which is correct, but they are not recording the patient as a COVID-19 patient. So the amount of information we are getting are actually very limited. So it makes a very wrong and misleading curve. So it's good, we can do the imaging, we can do the analysis and the lab tests, but we need to have a medical registry to register all the patients as COVID-19 patients.
Thank you. Thank you so much.
Thank you. Thank you, Professor Sanjay.
Nice to talk to you. Yeah, thank you.
Same here, likewise.
Yes.
Thank you, Dr.
Thank you.
Islam. I think our dean like to ask you about the recommendation for the future health crisis.
That's a very good point. That's a very good
Very big.
And if you allow me, this will take us again to the, it's not one size fits all, and it's not a single answer. There's a lot of actions that we would like to do, but we can actually summarize it in very few pillars. The first pillar is the healthcare system itself.
Yeah.
We need to have more beds. We need to have more people on board. We need to have someone who can actually predict. We need pharmacoepidemiologists in place. We need someone who can forecast future pandemics. The most actually shock happened to us in 2020 is that no one predicted actually the pandemic. If we do have the science of pharmacoepidemiology and the pharmacoeconomics, which is the science of forecast, we could have been at least more prepared for it. So first of all, we need to have a prediction research center. If we can record all cases, it will be great. We need to have more tests, more data. I always say it's more data in, more decisions, accurate decisions. Less data, less accurate decisions. So we are working in African countries because I not only work for Egypt data, I work with all the Middle East and African countries. And I can see that we have huge problem in terms of data. The less data you are giving me, the less insightful and the less decisions I can actually provide you as a health policy consultant. Second is we need to apply medical registries. It's very important to have medical registries. Anyone who's in home quarantine, anyone who's facing anything, the science of informatics is not well established in our healthcare systems. I've been working with the WHO and the CANREG, for example, the oncology registries. And you can see disasters in the oncology registers that physicians are not recording the cancer types, they are not recording the cancer sites. So we are faced with a mass awareness, lack of awareness in terms of physician level of the importance of informatics. Another thing is we need to have a committee between economists, between healthcare professionals, and health policy specialists. So if we are returning back to our opening of economy, we cannot do it at once. We need to have it planned. It can be planned only if we have a multidisciplinary teams in place. We need to have a good R&D and production. Luckily in Egypt, we are the first country in terms of production of pharmaceuticals and vaccines in Africa and Middle East. But we need to have a better plan. We need to plan our schools. We need to have governance. We are hearing a lot about the term governance, which is al-hawkama. And 2030 vision of Egypt is to have a complete governance. It means that I can control each government on its own. It's very important to say that Sharm el-Sheikh, for example, got 0 infections. I can reopen it and lock it down for tourism. However, Giza, I will completely make a lockdown because it got a huge surge in terms of cases. That's few of some solutions, but of course it needs a very integrated system between healthcare professionals, economists, the country itself, in terms of capacity, and the most important, the individual, us, to have a better awareness and actually to wear our masks. That's it, for example. I cannot hear you, Dr. Magda. I hope that answered your question.
Yeah, now it's okay. The internet is okay now. We heard you, and we are happy to see all this information, which is very important. Thank you, Dr. Islam. And now we will go back to our platform with Dr. Amin.
Thank you, Dr. Islam. And actually, by your presentation, we complete the puzzle, because we were asking during the pandemic, what did we miss? And we miss actually to have a data analysis according to scientific background to predict how far we are. And if we, for example, mention that we have 100 positive cases, we don't know this 100 from how many tested cases. So we can consider how big is or how risky is the situation is. And we would like to ask you to give the conference organization committee some recommendations to be included in our final recommendations. And I'd like to ask also all the participants with us now, you are most welcome to use the chat box to put on your recommendations for the organizing committee, because by the end of the second session, we will declare and announce the message of the conference, because this conference, as we have mentioned in the beginning and all the time, is for supporting healthcare heroes, and our support is scientifically support, is based on providing them by the science that they need to facilitate their mission in facing and tackling the situation, and also to be ready for coming crisis, not only in the second wave, but any crisis for human beings, especially the health crisis. Thank you, Prof. Islam. Thank you for the rest of us eminent speakers. Professor Yassin, Professor Mabrouk, Professor Femi, Professor Sanjay, Professor Islam. Thank you very much. And for the eminent professor who accepted to moderate this session, Professor Gamal Omran and Professor Magda Fathi. And for our participants, we have a break for 1 hour, and we will come back at 5:30 to complete our session and to listen to the winner of the best research in our conference, and also to listen to our message, our recommendations, and to have the group photo. Don't miss it, please. And we have more announcement about breaking news, but we keep it for the last minute of our conference. So see you within 1 hour. Thank you, Prof. Islam. Thank you for all the participants, and see you again.
To study the Arabic language since I was a kid. Reading about early Arab pioneers always made me feel super enthusiastic about this authentic culture. I got fascinated by the Arabic calligraphy. There was something always pushing me to study this language.
أول حاجة بنتعلمها في كلية إعلام إن صورة واحدة ممكن تعبر عن 1000 كلمة. أهم حاجة وإحنا بنختار الصورة لازم نتأكد إنها تستحق فعلاً إن كل الناس تشوفها. الكلية بالنسبة لنا مش مجرد مكان، ده عيلة وأصحاب وأحلى ذكريات.
Mon objectif, ce n'est pas d'apprendre une langue étrangère. Ce qui m'intéresse, c'est plutôt la communication avec les autres. Si nous parlons plusieurs langues, nous allons certainement mieux comprendre le monde qui nous entoure. Étudier une langue étrangère ne m'a pas seulement permis de comprendre les autres, mais aussi de me faire des amis et de trouver de nouvelles familles partout dans le monde.
Pengajian perubatan merupakan salah satu impian yang mampu dicapai di sini. Kami di sini bukan sahaja belajar tentang perubatan, namun kami juga belajar bagaimana cara untuk meraih keyakinan pesakit dalam menerima rawatan daripada kami. Harapan kami, semoga kami menjadi duta universiti ini sekembalinya ke Malaysia nanti. Tempat ini bukan hanya sekadar fakulti, tetapi ia merupakan rumah kedua bagi kami.
What does it mean to study here? one of the oldest universities all over the world, with a glorious past and promising present. 79 undergraduate colleges in different fields: science, pharmacy, astronomy, radiology, agriculture, architecture, engineering, art, sports, language, and Islamic studies. 46 research centers, 40,000 scholarships for foreigners, graduates from more than 150 countries all over the world. Welcome to Al-Azhar University.
African Academic Diaspora Virtual Homecoming 2020. The Association of African Universities, AAU, in collaboration with the Government of Ghana and the African Union, is inviting all educational stakeholders to the first virtual AAU Academic Diaspora Homecoming. The overall objective of the homecoming is to strengthen collaboration and partnership among research and education institutions of higher learning in Africa and the African diaspora to advance quality of education and general well-being of all persons of African descent. The main theme for this event is Advancing African and Diasporan Academic Relations. The date is September 28th to 30th, 2020. Time is 15 o'clock GMT. Kindly convert the time to your local time. The venue will be the Virtual Association of African Universities e-Forum, with the registration link on your screen. For more information, contact us now, namo@aau.org or secgen@aau.org. You can call us or WhatsApp us on +1 312 539 8473, +233 243 298 464. African Academic Diaspora Virtual Homecoming 2020, Advancing African and Diasporan Academic Relations. African Academic Diaspora
present in Scotel area.
System, now it's very important to have a good supply chain system and to do a centralization of decisions. As we seen in the first of pandemic, it was a Ministry of Health decision, then it was a Prime Minister decision, and now we can see that all the presidents all over the world are the one taking responsibility of the system and for COVID. So that's need to be planned and defined for any future pandemic. Should I go for R&D or just production of repurposed drug? The process, lockdown scenarios, having a hotline in place, awareness campaigns, and establish a public-private partnership. So these will summarize for Egypt specifically 10 steps to mitigate the risk of wave 2, increase number of tests, making it proactive, apply medical registry for all home quarantine, doing quantitative assessment, not to open all